Peace be upon you. Assalamu alaikum. In this video, I'm going to talk about this subject, autoimmunity. I'm going to define the process of autoimmunity, the two types of autoimmune diseases, defining the process of tolerance, learning how genetic and environmental factors interact or predispose uh, the processes that lead to breach intolerance, and talking a little bit about uh, diagnosis and treatment strategies. So what is autoimmunity? It's a pathological condition identified or defined by abnormal autoimmune responses mediated by autoantibodies and T-cell responses to self-antigens. So in other words, it is failure to host immune system to distinguish between self and non-self antigens, so lead to destruction or our tissues and organs. So in early 20th century, Paul Ehrlich defined it as horror autotoxicosis when he realized that sometimes immune system can go awry, and which result in a spectrum of clinical syndrome. Such diseases affect 5 to 8% of human population, so they are often chronic and require prolonged medical intervention at least to relieve symptoms. So as a summary, it's a multi-step process, require many predisposing factors such as environmental factor, genetic factor, epigenetics. When the disease is initiated, so step by step or as time passes, it leads to tissue destruction, especially when it is not treated. Finally, it will damage the organ, and the destructed tissue should be replaced by scar formation, so which may lead to organ dysfunction. Autoimmune conditions present a spectrum between organ-specific and systemic diseases. For example, we have here Hashimoto thyroiditis, so we'll talk about it. In organ-specific, the cell of the target organ may be damaged directly by immune cell infiltration, antibodies, T cells, or indirectly. So, for example, antibodies may overstimulate or block the normal function of the target organ. So, in Hashimoto thyroiditis, some autoantibodies target thyroglobulin and thyroid peroxidase, which exist in this thyroid tissue or thyroid cells. So they aid in iodine uptake. So when the process is disrupted or interrupted, the process of iodine uptake, which is a very important substance for the thyroid function, when the process is interrupted, it leads to hypothyroidism and may also cause thyroid hyperplasia, and secondary to immune cell infiltration. So this is about Hashimoto thyroiditis. We have other organ-specific diseases such as multiple sclerosis, which affect brain and spinal cord, or which affects central nervous system, or generally it affects nervous system. It's a demyelinating disease. Hashimoto thyroiditis, thyrotoxicosis or myasthenia gravis affect the junction between muscles and the neurons. Pernicious anemia, which affects stomach, and Addison's disease, pancreas, insulin-dependent diabetes mellitus. However, about systemic diseases, the immune response, it's not restricted, or it's not specific for an organ, but it's directed to a broad range of target antigens and involve a number of organs and tissues. So the damage is typically widespread, and it could result from either direct cellular damage caused by antibodies or by accumulation of complexes, such as in systemic lupus erythematosus. So there is a question, so what lead to this difference between organ-specific and systemic autoimmune diseases? So basically, it could be many reasons, but one of the most important is the location of the antigen. There are some antigens which are specific to an organ, for example, which exist in thyroid or in pancreas, that they doesn't exist in other sites. Oppositely, or in contrast, there are some antigens. It's possible that they exist in all or most of human body cells. So the example of these antigens or widespread antigens is the proteins that associated with some cellular process such as cell differentiation and cell multiplication. For example, the proteins that regulate cell division and the proteins associated with the, for example, translation, and others which are associated with DNA. This, because DNA exists in all cells, so the proteins also exist. So when there is a damage, for example, by an inflammatory process, by a trauma, so which may lead to inflammation, by an infection, it lead to the spread of these antigens to bloodstream or to out of the organ, which may stimulate or induce human immunocytes or immunocytes, then are recognized and are displayed on MHC to T cells, which later lead to response. The systemic diseases are dermatomyositis, SLE, scleroderma or skin scleroderma, and joint rheumatoid arthritis. So what is tolerance? Tolerance is a process in which human immune system recognize or differentiate between foreign antigens and our body cells, or our self-antigens. So it's a process by which human tries to stop or prevent any interaction between immunocytes or cell reactivity to our self-antigens. So it's basically done by several processes. For example, as we see here, antigen sequestration, we'll talk about shortly, and then later we will give detail. Central tolerance, which occur in bone marrow and thymus. Then there is peripheral tolerance, which occur in secondary lymphoid organs, and by using of T regulatory cells. So what about antigen sequestration? So basically, there is some organs or some antigens in our body which form a very delicate organs, and for example, central nervous system, testes, ocular tissue. These sites, especially CNS, they are both delicate and have very low replicative potential, so they don't divide easily. once they are formed, for example, when there is a damage to the body of the cell is destroyed and it will never get back. So in these organs, during T cell and B cell development in bone marrow and thymus or primary immune organs, these antigens are not displayed to them because immune system or T cells are very active for killing cells. So it's very easy to them to kill a cell, even if it's our cell. Even these organs or upregulate fast and fast ligand. So the benefit of this fast and fast ligand is that whenever a lymphocyte is activated, so they have their half-life. It should undergo apoptosis. So when they, for example, one pathway of this apoptosis is engagement of fast and fast ligand. So it leads to the death of the cell. So this is antigen sequestration. This is a method to prevent reactions of immune system with our body cells or antigens. Second pathway for providing tolerance is central tolerance. About central tolerance, so there is three processes happen which are clonal deletion of autoreactive lymphocytes, so it's called negative selection, B cell receptor editing. It's a very special process for B cells. Some evidences say there is it exists also for T cell, but it is not significant, or so not much papers indicate that. And generation of natural regulatory T cells. So what's negative selection also? During T cell and B cell development, because of their receptor rearrangement of the genes, for example, VD and G, gene recombination to produce a B cell and T cell receptor diversity, it has a potential to produce autoreactive T cell and B cell receptors. So during their development, they should undergo testing to see if they could react with self-antigens with high affinity. Simply, they should undergo apoptosis at there. As we see here, it's true for both T and B cells. For B cells, there is another chance to them. They are sent to rearrange their receptors. Simply, they are sent to rearrange their receptors, B cell receptors, to produce receptors with much lower affinity, or they should undergo apoptosis too. So the cells with very low affinity for self-antigens are led to go to the bloodstream. So what about cells with medium affinity? Simply, some kind of regulatory cells are produced. They are called natural regulatory T cells. So the central tolerance for T cell and B cell, clonal deletion of high affinity T and B cells, receptor editing for B cells, and generation of natural regulatory T cells. We talked why all antigens are displayed except for CNS, ocular, and testis antigens. answer this question because these organs are very delicate, or it could be due to the very low replicative potential of these organs or tissues. So that's what happened in central tolerance. What about peripheral tolerance? So before we get to there is a question why central tolerance was not sufficient, because despite that process of central tolerance, that rigorous mechanism, there is still some autoreactive lymphocytes that can escape and go to bloodstream. So there should be another process to prevent the potential damage which may lead to autoimmune disease. And there is a process occur outside of primary immune organs is called peripheral tolerance, which occur in lymph nodes, spleen, gall, smalls, and so on. So in this process, B cell and B cell, for example, when a B cell and a T cell are autoreactive, they are induced to be anergic or to be unresponsive. This happened due to lack of co-stimulatory signal provided by antigen-presenting cells. B7 ligand, it's called. Activation induces cell death, as I talked earlier. Whenever a T cell is activated or a lymphocyte is activated, it has a very short half-life and it should go on death. And it should express fast and fast. Like when these receptor and proteins and ligands are engaged, the cell undergo death. And there is also producing regulatory T cells in periphery, as it's called induced regulatory T cells, through upregulation of FOXP3. So this gene is associated with, or it can regulate many other proteins associated with the regulatory phenotype of these T cells. In periphery, they should also, some of the autoreactive should also go under death, or when they are activated, finally they should die. Some of them should become anergic, so it will remain unresponsive to the same antigen, and some of them will be induced T-regulatory cells. So we have two types of regulatory T cell: natural regulatory T cell and induced regulatory T cells. So the natural regulatory T cells are thymocytes with immediate affinity, intermediate affinity of antigens encountered in the thymus. So they upregulate the transcription factor FOXP3 and simply become natural regulatory T cells. We have another class of regulatory T cells, but they are produced in periphery, not in thymus. They are called induced. Some naive T cells that have escaped to the periphery can also be induced to express FOXP3 and acquire their regulatory function. There are some factors that may induce this phenotype of cells, which are presence of certain cytokines, for example, transforming growth factor beta, chronic low-dose antigen exposure, lack of co-stimulation, or presentation of antigen by immature dendritic cells. These all lead to formation of induced regulatory T cells. We talked about this. As we see here, So we talked about peripheral tolerance, about the regulatory T cell. So now we will talk about energy, how a cell become energetic. We talked about when there is no co-stimulatory molecule. Yes, as we saw here. For example, antigen presenting cell, and this is a T cell. Suppose that this is the co-stimulatory molecule on the T cell, but the co-stimulatory molecule doesn't exist on the antigen presenting cells. So this cell become energetic. Or in another case, there is co-stimulatory molecule, but there is no engagement with the MHC. So there is no antigen presented on the antigen presenting cell. It has no effect on the T cell. However, when there is both MHC plus peptide with co-stimulatory molecule, it leads to the activation of the T cell. So this is how the cell become energetic. When does this happen? We said a presentation of antigen, for example, by an imager antigen presenting cell, or presenting self antigens by cells other than immunocytes. This process happens naturally in our body. Or how B cells actually become unresponsive. As we know, one of the reasons is chronic long exposure of a soluble antigen without co-stimulation. As we know, B cells, for their activation, require CD4 T cell help. We know that a B cell can recognize an antigen for itself, but it can be activated unless there is a CD4 T cell help. So there should be a specific T cell for the same antigen that has been recognized by the B cell. There is some T-independent antigens, but they are very low in number or in human cases. So for generally, it needs T cell help. For example, when there is no autoreactive T cell, even if the B cell exists, it can't get activated without T cell help. So that T cell, there is no signal, and the B cell become unresponsive. This is how the B cell become energetic, and this is the normal process for B cell activity. As we see here, there is MHC plus peptide. There is a T cell receptor engaged with the antigen presenting cell. For example, if we suppose the T cell is activated, it displays, or it can recognize the same antigen displayed by B cells. Now, when the B cell is activated, it can send signals to tell the B cell, yes, become activated. We need to mount an attack to this antigen. This is the normal process happen, but here the process how it become energetic. So till here we talked about tolerance and the process of positive and negative selection, the regulatory T cells, and energy. So now, how T regulatory cells lead or regulate immune response? There is three basic effects or three basic pathways. First, they can kill antigen presenting cells or effector T cells directly by releasing granzymes and some other cytolytic proteins. Secondly, they can upregulate another protein which is called CTLA4. It's instead of CD28, which exists on activated T cells, but CTLA4 is an immunomodulatory protein. It exists on T regulatory cells when it is engaged with the CD8 or CD8 and 86 or B7 ligand. It causes the reduction of the co-stimulatory molecule, reduction of pro-inflammatory cytokines such as interleukin-6, tumor necrosis alpha, and even the antigen-presenting cell itself will produce some inhibitory factors, for example, indolamine 2,3 dioxygenase. So it produces an immunosuppressive microenvironment. The third mechanism is that T regulatory cells express only low affinity interleukin-2 receptor alpha, so CD25. It's just like a sponge. It can absorb the cytokines around and with no much effect. So it will cause the reduction of the cytokines in the environment and produce an immunosuppressive environment. There is another process it can do. It is that called linkage suppression. For example, when a T regulatory cell is engaged with an antigen-presenting cell for peptide A, it will cause the deactivation of the effector T cell for the specific for peptide A. The same process here will happen here, but at the same time, for example, when there is another specific T cell, for example, for peptide B, for another antigen, engaged with the same antigen presenting cell which interact with the T regulatory cell, it will cause the deactivation of this T cells specific for antigen B. So this is called linkage suppression. This is very interesting. What are the factors that favor susceptibility to autoimmune disease? So one of the most important is genes related to MHC haplotype. So this is one of the intrinsic factors. For example, before we jump to there, the odds ratio approximates the value of increased risk of the disease associated with inheritance of a particular HA allele. So, for example, inheritance of these alleles increase the risk by 4 times or by 12 times. For example, Hashimoto's thyroiditis tend to be associated more with the DR5. The shared epitope is also present in the heat shock proteins of various bacilli and Epstein-Barr virus, GP110 protein, and this is a microbial cross-reacting epitope with anti-citrullinate antibodies of rheumatoid arthritis. So this is about shared epitopes. HLA-A1, BA, DR3, also they are linked to SLE and so on. So certain MHC haplotypes are associated with or at least they increase the risk for certain autoimmune disease, as we see in this table. So in previous videos, we have talked about MHC, but the exact mechanism how MHC leads or predisposes to autoimmune disease, it's still unknown. There is polymorphism in non-HLA genes, for example, at first complex gene interactions, for example, PTP, N22, which is involved in signaling in many immune cell populations, and it is the polymorphism in this gene is associated with these diseases, which is RA, type 1 diabetes, and autoimmune thyroiditis. Node 2 is a cytoplasmic sensor of bacterial peptidoglycans, so the polymorphism in this gene is also associated with Crohn's disease. The problem in complement proteins is also associated with many diseases, as one of the most important functions of complement proteins, despite its immune functions such as producing pores by membrane attack complex on microbes, another function, it helps to opsonize the antigens so as to be cleared by other immunocytes, for example, a macrophage. When there is a problem, for example, deficiency in these complement proteins, the clearance of antigens is deficient, so the antigens persist in the site of the damage, which may lead or amplify the immune response. Interleukin 23 receptor, this is associated, or its effect is variable according to the mutation, as this is associated with T helper 17. And T helper 17 is also associated with many autoimmune diseases. The receptor we just talked about, interleukin 2 receptor alpha, is the polymorphism here is associated with multiple sclerosis, type 1 diabetes, which is linked that the change in this receptor may affect generation or function of T regulatory cells. There is simpler genes and which are called Mendelian abnormalities. For example, problem in this gene, ARE, which is associated with the established mechanism of central tolerance, will lead to autoimmune diseases, and this also FOXP3, interleukin 2 receptor alpha, CTLA4, Fas and Fas ligand. All these lead by some mechanism which require much detail, may lead to autoimmune disease. For example, this lead to death of activated lymphocytes, or especially T cells, and these are inhibitory or anti-inflammatory cytokines. There is extrinsic factors which, for example, could be infections. For example, some bacterial or viral antigens could mimic our self-antigens, which may cause a cross reaction, or even sometimes they may cause direct organ damage and release of self or some hidden peptides, which may lead to the injection or the process. Toxins, smoking in rheumatoid arthritis, which could modify some antigens and may initiate the process. Iodine is also associated, fabric dyes, scleroderma, stress, ultraviolet, which may lead to release of nuclear content of cells. This is associated with SLA, and it's very significant. And foods, for example, gluten, breastfeeding cessation or giving the infant of cow's milk, vitamin D deficiency or obesity, exposure to pesticides, heavy metals, and all about geographical area. Also, it is very, very significant. So all of these factors are linked to increased risk of autoimmunity. There is one other important reason, is gender and hormonal differences between male and female. As we know, the incidence or prevalence of autoimmune disease is much higher in females than males, and this is actually related to many reasons. For example, hormonal, the genetic content. For example, there is some hypothesis such as the process of X chromosome inactivation, disbalance, many things. But the hormones also have many or have a prominent role, especially progesterone, estrogen, and so on. So how the process is breached, how the tolerance is breached. For example, activation of antigen-presenting cells by an infection or inflammation. We said previously, or in previous slides, we said in humans, there is a very, despite that it's low, but they exist. So the autoreactive cells exist, but they require co-stimulation. So when there is an inflammatory process which may activate antigen-presenting cells to upregulate co-stimulatory molecules, when these autoreactive lymphocytes engage with an antigen-presenting cell expressing co-stimulatory molecules, simply they are activated and the process has started. As we see here, there is a B7 ligand and CD28. Molecular mimicry, for example, whenever there is some antigens in our body which are mimicked by microbial antigens, this may lead to cross-reaction and again autoimmunity. In some cases, some kind of antigens they don't undergo the conventional process we have learned of antigen processing and presentation. They directly bind to a groove between MHC and T cell receptor, leads to unproductive activation of T cells. In them, there could be autoreactive T cells. So they do that by cytokine storm. So as I said, there could be some autoreactive lymphocytes could be activated and may lead to the initiation of the process. Anatomic alterations caused by caused by inflammation, for example, the areas we talked about in CNS, for example, or in eye, when there is a traumatic damage to one eye, it caused release of previously hidden antigens or sequestered intraocular protein antigens which have not been displayed to developing T cells in the thymus. So they simply don't know these antigens exist. They presume them as foreign. So simply when there is a attack or antigen exposure from one eye and the stimulation of lymphocytes, the damage of one eye, which may lead to the destruction of the other eye. There is another mechanism, which is the waste disposal hypothesis. Especially in SLE, there is the process of clearance of antigens is very it's a very bad. It is not done in a conventional way. So the exposed antigens to the bloodstream, they are not cleared efficiently. For example, they could persist for more time, phagocytosis, and displayed to lymphocytes, which also this may lead to the injection of production of autoreactive T and B cells. So when I say B cell, I mean auto antibodies. Despite that, autoimmune disease is a multi-step process, so it doesn't happen very easily. There should be, the most important is that there should be genetic predisposition. There is some, as we saw earlier in previous slides, some, for example, HLA haplotypes are associated with some type of disease, or they are linked, or they increase risk of some disease. Some other haplotypes with other disease. So there should be the genetic content or genetic susceptibility. In addition to that, they need other triggers. For example, environmental agent, as we talked about, it could be a bacteria, virus, a toxin, metals, and so on. So when these all exist, then it may lead to process of autoimmunity. Also, antibody production alone does not equal to autoimmune disease because, for example, in about 8 to 13% of healthy humans or human population, they have some kind of autoantibodies, for example, some kind of anti-nuclear antibodies. This is, however, they haven't the disease. So antibody production does not equal to the autoimmune disease. People with autoimmune disease, they could have more than one disease. For example, many people with thyroiditis or the autoimmune thyroiditis, they could have problems with GI tract. For example, they could have celiac disease. So it's a very complicated, it's not easy to produce autoimmunity. It's also occur in stages, so it occurs progressively. For example, at first there's the predisposition, as we talked about. There should be the genetic and environmental predisposition. There should be an initiation process, so there should be something lead to inflammatory process and organ damage, or the allocation of the antigen and injection of immunity. There should be progression in which the auto or the inflammatory process is heightened by exposure of more antigens. It's just like an amplification process. In final stage is propagation, or sometimes also it's called amplification. For example, we have some conventional antigens. Antibodies, for example, are produced for, but in later stages, the antibodies can recognize some kind of epitopes which were previously hidden. And this is by the continuous inflammatory process lead to formation of different types of epitopes located in the these apoptotic cell bodies. This process is called epitope spread. So about the diagnosis, each of the disease is related to a spectrum of signs and symptoms. Their diagnosis is linked to asking doctor to do many laboratory tests, for example, antibodies, and citrulline-mediated antibodies in rheumatoid arthritis, anti-nuclear antibodies. They exist in many people with SLE. They are sensitive but not specific, so anti-thyroid peroxidase, anti-thyroglobulin, and so on, they are associated with the autoimmune thyroiditis or Hashimoto and other types of thyroid diseases. Some kind of autoantibodies to, for example, insulin, or anti-GAD. Many other types of antibodies have a very important diagnostic value for these autoimmune diseases. About the treatment, some treatments are general. They induce immunosuppression generally. There are some trials which tend to deactivate some processes in the inflammation or some kind of specific cells in the autoimmune disease. Until now, they have not been much effective, and the treatment aids to relieve symptoms and produce a good quality life, as good or as best as possible. So we join the end of our meeting or our subject. If you have any question, you can comment, and these are the references.